with that, we're going to start talking about peptide bond formation. Now, this is literally the amino acids coming together to form proteins. And in order for it to officially become a protein, you need to have more than 50 amino acids linked together. And sometimes amino acids come together in the body to form other functions with 2 or 3 or 4 amino acids. So amino acids can come together in a peptide bond to make smaller structures, but they can also make large structures like proteins. So this is the, I'm going to activate the pen. Pen is activated. So we have an amino acid right here, and we have our amino group, and we have our carboxylate group. This happens to be glycine because we have a hydrogen here, and then we have the hydrogen here. So the hydrogen is actually the R group. So if I just kind of erase that there. So we've got glycine coming together with another glycine. So our carboxylic acid group and our amino group, the OH group from the carboxylic acid group comes off, and the hydrogen from the amino group comes off, and they link together to form peptide linkage. And a byproduct of this is water. So I have 2 amino acid residues here. This is a full amino acid, and this is a full amino acid. Now, a residue is something that is left over. It's what's left over after something is done. So in this case, the residue is what's left over after the peptide bond has occurred, or that peptide linkage. So it's losing something. It's losing one of the amino acids is losing an OH group, and one of the amino acids is losing a hydrogen. So if it's losing something, then this is what's left over. So here we have 2 amino acid residues, and we would call those glycine residues. And this would be called a dipeptide. Now, this is, it's also called an amide bond. An amide can be recognized by, within a hydrocarbon chain, this pathway here that I'm following with my pen is what makes up the backbone of proteins. And within that, at that peptide linkage, we have an amine here. The amine can be recognized by a carbon attached to a nitrogen within a hydrocarbon chain, and that carbon has a carbonyl group attached to it. So a peptide bond, peptide linkage, also known as an amide bond or amide, depending on who you are talking to and how it's pronounced. You may hear me pronouncing it both ways. So this is called a dehydration synthesis reaction, and it's because in the process of these amino acids losing some of their stuff, one amino acid loses an OH, one amino acid loses a hydrogen, and they come together to form water. So dehydration, it's kind of like we're removing this water from the amino acids, and we are condensed. So a condensation reaction is, like on a cold aluminum can in the summertime, you see condensation on the side. It's like the formation of water on the side of the can. So in here we have the formation of water with each peptide linkage. So here is that reaction summarized again. So an OH group from one amino acid hooks up with hydrogen from another amino acid. And in my presentations, I have always preferred this orientation of putting the nitrogen, this amino group, on the left-hand side, And there's a reason for that, because in many of these not animations, but examples, this is the way it's shown. And this peptide linkage shows the amino group on this side with the carboxylic acid group on this side, and it is oriented so that it's facing the amino group right here on this other amino acid. So this amino group loses its hydrogen. Here we have the peptide linkage with that amide bond, peptide linkage, and the production of water. So if we were to destroy this protein and separate the two amino acid residues from one another, we can do the opposite in a hydrolysis reaction. Lysis means to cut. If we add water under certain conditions, very specific conditions, to a peptide linkage, we can split those back in two and turn them, the amino acid residues, back into their amino acid. So that is one of the ways we're going to learn that proteins can become broken down back into their amino acids. So if we have a dipeptide, we will have one water molecule. If we have a tripeptide, 3 amino acid residues, there will have been 2 water molecules produced. A tetrapeptide has 4 amino acid residues. When those 4 amino acid residues came together, so let's say 1, 2, 3, 4, we have 3 total peptide linkages, which means that 3 water molecules would have been the result of the formation of this tetrapeptide. A pentapeptide has 5. Polypeptides contain between 40 and 50 amino acid residues, and proteins are longer than 50. So there is a way that we can represent peptides, and in a sequence of amino acids here, we have the 3-letter code for each of the amino acids. And in this sequence of amino acids, if we were to draw it out, the NH2 group would be free at this end because it was not involved in a peptide linkage. And at this end, I keep having to move my picture here. At this end, we will have a free carboxylic acid group that was not involved in a peptide linkage. So it still has its OH group. And this amino group on this end still has its hydrogen. So a way that we represent this is we call this the N-terminal residue. The N-terminal residue is the amino acid that is first in this sequence. The C-terminal residue is the amino acid that is the last. So when we talk about protein synthesis, which is a product of reading our DNA genetic code, which tells us what order these amino acids need to assemble in, the very first protein that comes out of a ribosome, the very first amino acid that will come out of a ribosome will be the N-terminal residue. And the amino acids are sequenced after that. The very last protein that comes out of the ribosome, and when that protein is done being formed, that is called the C-terminal residue. So if we look at a protein chain, there's a protein chain. We can often refer to this end as the end terminus. And I want to do some color coding because you know how much I like that. So the N terminus is often represented by a blue color. And the C terminus is often represented by a red color. And that goes back to these, it's beautiful. That goes back to this common color coding that you see with blues associated with bases. This is our nitrogen end. Nitrogen becomes gets basic properties. So I think that's why we're using similar colors. So oftentimes in protein models, you will see a blue end and a red end, showing the beginning of the protein chain and the end of the protein chain. So the C terminal residue is found at this end, and it's written on the right. So if you see a sequence of amino acids like this, and I ask you for the N terminal residue, you want to start with the one on the left. And if I ask you for the C terminal residue, you want to start with the one on the right. So I think I'm going to end this video talking about, so just shorten it a little bit. So the focus of the video has simply been polypeptide formation and the processes that go into bringing amino acids together to form peptide chains. And then in the next sequence, the next video in this sequence, I'll talk about protein structure. So with that, I'll see you at the next one. Thanks for watching.